I think I've showed you this slide already, this idea of, plants are just phenomenal in the sense that they can regrow themselves over and over and over. And even though you leave, either through predation or mowing your lawn or whatever, that you take a large part of the plant away and it can still come back. And the reason for that is the decentralization. They don't have a specific place for a heart like we do, or a brain, or a liver. Everything's decentralized and spread out over the organism so that you can really chop them up amazingly, and they can start to regrow into a full, complete plant. And so that knowledge has been known for quite a while, but then getting, the lab techniques to figure out how to capitalize on this and work with it. And so the fancy word for this is totipotency, which just means that you can grow a full plant from just a handful of plant cells, and that's all you need. And it, as long as you understand how to work with the plant in the lab. And, even if you work with a mature plant, you can take part of the leaf tissue, just a little chunk, and you can get that to grow into a complete plant. Even though you started with a mature plant, you think, oh, well, that leaf is always going to be leaf tissue. But no, because they can do a tremendous gene expression shifting if you give them the right environment. And so you can take a little tiny chunk of the leaf tissue, put it onto a certain media that has the right ingredients in it, and you can coax that little chunk of tissue to become the mature plant again. It'll be a clone of the plant you took it from. It'll be exact same. You didn't go through any kind of sexual reproduction. So here you're seeing kind of different stages. This one is with banana. And this is tissue culture, where you have a special agar media recipe that's similar. You remember when you did your soil isolation of microbes and you worked with agar in agar plates? So it's a similar idea. It's just that you create a recipe that will favor the growth of these plant cells. And so this is a classic. We used to do carrot tissue culture quite a bit, plant fizz early on. But I've been trying to switch it out to some other plants too, but carrot works really well because you can go to the store and buy carrots. And after you wash the carrot and surface sterilize, you can cut the carrot and then just work with some of the inner tissue. You take a core of the inner tissue, which is pretty safe because you're not taking anything on the surface where there's still surface microbes hanging out and could cause problems. So you go into the inner tissue, take a core, put it down on a plate or a sterile surface and make cuts, and then you transfer it to an agar media that has tissue culture recipe in that with certain hormones, and you can coax it to form. Usually what we do is we get it to form a callus first. So I'd like you to know that word, please. So a callus is this idea that you have these, in this case, the carrot cells are coaxed through some hormones in there to divide, grow, divide and grow, divide and grow, but they're not differentiating yet. They're not turning into root tissue or leaf tissue. So it's a callus. It's undifferentiated growth is essentially what we're talking about. And then often what you do is you take chunks of that callus. You don't even have to take the whole callus. You just take a subculture of it and put it onto a different recipe of tissue culture media that has some slightly different hormones. And that triggers the callus cells to go ahead and start differentiating. And so they'll start to grow roots and shoots and leaves until you have a full mature plant that's a clone of the carrot plant you started with. So carrot works really well because you can often get really high results with no contamination, really good results. And that's mainly because you're taking interior tissue. The one thing you don't do is take smack in the middle, because that's where the vascular tissue is. There's a lot of dead cells there. So you don't take smack in the middle. You try to go around. But that works really well. And so this idea that you're taking just a small piece of the plant It can be leaf, it can be taproot, like you just saw with the carrot. It could be root. Often root tips are used because the apical meristem, the root apical meristem is very active. So if you take a root tip or a shoot tip, so pretty much any part of the plant can be used. And the biggest thing here is that you need to try to surface sterilize the plant. If you're working with a leaf tissue or the carrot, you have to knock out any of the microbes that are on the plant. And that's probably the hardest thing that we face when you do tissue culture, because you have to kind of troubleshoot that process and figure out how much surface sterilization is needed, how aggressive do you get with the chemicals. So that is still kind of varies plant to plant. Some species are really sensitive. Usually we use alcohol and bleach to surface sterilize. And sometimes that, if you let it sit for too long, it causes damage to the plant, or it doesn't sit long enough and you still get massive contamination when you start to try to grow the plant on the tissue culture medium. So sterility is a big problem and probably the biggest thing that you have to overcome to get this to work. But the goal is to get those cells to divide enough that you get a clump of cells that you can work with. And then, like I said, you can subculture that. You can take many clumps of that callus and distribute it into fresh media. And so you can see, if you can imagine that, you don't need a ton of material to start with, really, just a little bit of mass of cells. And then you can grow a bunch of plants from that. Not only that, but you're growing them in these little tiny Petri plates or little tiny vessels. So you can grow a ton of plants in a very small footprint. And so commercially, that's very attractive. If you can get plants grown up in a very small space, you don't have to worry about, big pots of soil. You're just using the tissue culture medium in these small containers to get these plants to grow. And then eventually you can pot them out or sell them. So, whoops, sorry. This idea that you can coax these plants to form a clone and become a mature plant from just a bit of tissue has been, like I said, known for a while, but trying to get this to work for all the different plant species, there's been a lot of troubleshooting and a lot of failed attempts until they can figure out what's the right procedure. So there's a lot of initial work that goes into this, but then once you have a procedure down, you can mass produce plants really readily. So a huge number of plants can be generated, and they, like I said, pretty much any part of the plant you can pick. You just don't pick vascular tissue that has a lot of dead xylem cells in it. Don't do that. But when you make the media, the tissue culture medium that you grow these cells on, you usually just provide the minerals that a plant needs that normally it gets from the soil system. And then you often will put hormones in there too. And you have to know the right ratio and amounts of hormones to get them to grow, and a little bit of sugar typically to get them a little bit of nutrients as they're starting their growth. The type of hormones that are, we talked about this way back in plant growth section, that auxins and cytokinins are the go-to hormones. There's a lot of different types of auxins and cytokinins, so they pick slightly different ones depending on the plant, but they'll pick a ratio of those in order to have them mixed into the tissue culture medium. And then some recipes, if you look at it, they'll use things like coconut milk or yeast extract. And that's also to do with the idea that those types of ingredients do have some minerals in them, but they also have some natural auxin hormones and cytokinins that can be used. So sometimes you'll see recipes that call for things like that. But whatever recipe you follow, basically you want minerals in there, you want hormones, and often a little bit of sugar. And then, of course, you add that agar to get everything to thicken up and gel. And then you pour it into either a Petri plate or these are called magenta vessels. It's just a little square box that's capable of being autoclaved and sterilized. And you can see the little plantlets in here growing, and you don't put a ton of agar in there because you want the roots to be able to grow nicely into the gel of the media. And so you can see that nicely in this picture that they're growing in to the gel and getting their nutrients that way.
Gel nutrient, like plant dependent.
Yes.
Different recipe.
Yeah, in general, well, there's like universal, often they just call them mineral salts medium, because in general, all plants need the same minerals pretty much.
Yeah.
There's a few exceptions, but,
Okay.
and then the hormones though can vary a little bit. So sometimes you'll add those in separately. But very commonly now you can buy tissue culture media, like a one bottle that has everything in it. And you just mix that up and it works. So this, I guess, actually, not just pathogens, that's probably not the best choice of word, but pathogens and just in general, microbes, okay? So, I can't get it to show up. So microbes, basically this idea of you need to create a plant that's pretty much sterile to get it to grow successfully, because this is a very artificial system. So if there's any microbes on the plant that didn't get killed off in your surface sterilization, they'll take advantage of the mineral or of the nutrients in that tissue culture medium, and they can often overwhelm the growth of the plant. And so you end up with, you'll look at your vessel, several days later, and it'll just be swarmed with microbes, not necessarily pathogens. It could be some pathogens in there too, but just in general, it can get out of balance like that where the microbes just take over. And so that's the battle you're constantly trying to fight and get that to work. And here, we use the word plantlets when we start to see the full plant forming here. That's a common term that you see. So here is an example of with cannabis sativa, and they're looking at, how can we do tissue culture with this plant, and you could see the pictures. Some don't look so good here. So you have this idea of anything that you see here, this white fluffiness or this kind of tan sliminess. Yeah, isn't that cool looking? That's probably a bacillus species, if I'm guessing, but rather than a slime mold, but they can look like that. They really weird, almost looks like nerves in a brain or something. But you can see the plant is not doing well. Okay, so this is what I mean by massive contamination. It can just overwhelm. However, this one, see, this one's still looking pretty good. Yeah. And it has some modest growth. So I've seen this over and over, and I'll explain in a minute what I think is going on here, but like this could be a success story. Like this could get transferred into another clean vessel and it could make it. The rest, not so much. They're pretty much toast. And so this is what you're always trying to keep an eye on and assess and keep your fingers crossed and, do a little lucky dance that everything's going to work out, because sometimes.
Is there a chance that it could be like symbiotic, that the microbes can help the plant, and maybe that's why it's looking so good in the
Yep, you're right. And so I'm getting into this idea in a sec. You're right on track here. So let me just show you this next slide. So here are, I think this is carrot tissue culture, where they took, they surface sterilized the taproot, and then they took some chunks of the inner carrot. And I just wanted to show you, so this is a tissue culture plate. And so everywhere you see the carrot, you can see that there is, except maybe this one is probably the cleanest, but here you can see lots of microbial growth. And then you also see microbial growth away from the carrot chunks as well. So a couple things are going on here. The one thing you can't control, well, you can control it, but it's, again, a lot of troubleshooting. There's microbes that hang out inside the plant, okay? And I think I've talked to you maybe about this before. The fancy word, does anyone remember? Endophytes. So in the carrot taproot, in whatever you're using, there's endophytes. You know, we are well colonized with trillions of microbes in on us. Plants are the same way. They're not living in the plant cells, but they're in the tissue of the plant, all right? And they're very beneficial. We're learning more and more about what the microbes do for the human, what the microbes are doing for the plant. And by and large, the ones that coexist with the plant long-term are helping the plant. It's a win-win. And so these endophytes are there. You can't kill, like when you're trying to surface sterilize and you're like, I want to get rid of everything, you can't get rid of the endophytes because then essentially you kill the tissue of the plant too. So there's no way to effectively kill the endophytes in that regard. However, you could put certain antibiotics or, well, yeah, antibiotics into the media to retard their growth so they won't overwhelm the system. And I've tried that too, and that can be met with some frustrating results because I was doing that with African violet. And the African violet plant didn't like the antibiotic and died. And so I got really mad because it was for Plant Fizz several years ago and everyone's plant died. So I called the company and I said, you told me this would kill off the microbes, but it's killed the plant. Oh, yeah, sometimes that happens. And I'm like, thank you. And so, you know, a whole batch of, 48 students, plants dead. It was just so frustrating. So there's that risk. If you do put in antibiotics, you first have to test the impact on the plant, what dose. So what you have happening here, the ones that are coming out or around the tissue here, they've grown out from the carrot taproot and are seeking the nutrients out into the agar. So these are endophytes, the ones that are growing out. But the other ones that are far away from the chunk, those must have fallen in to the plate when they took the lid off and there was, wafting of air current, because there's a bunch of stuff in the air that we're, being exposed to, all kinds of microbes and microbial spores. And so they landed there and grew. And so just by examining the plate, you can tell the difference typically between endophytes and a contaminant that fell in from the air. The other idea too with endophytes is that in general, the rule of thumb is if you do this experiment where you put the carrot chunks down and then you go and incubate it, if you don't see anything growing, any microbes, within the first 48 hours, but then you start to see things popping up after 48 hours, that's typically an endophyte, especially if you see it growing out. So the first 48 hours is kind of crucial that you keep an eye on it so that you can kind of assess what's going on. So the reason we say 48 hours, some people say 24 to 48 hours, is it takes a little while for the endophyte in the tissue to work its way out. It's got to get out and onto the agar and then start to grow well enough that we can see it with the naked eye. And that usually takes a couple days. But if you see things popping up right away, that's usually a contaminant that we don't want. And so going back to this picture, this plant, I'm guessing these are endophytes and they are keeping each other in check well enough. And I've had this situation many times where then I can take the plantlet and put it into a fresh media and it goes fine. And so sometimes it can be rescued. It just depends on the situation. The rest of those, I would say no. Even if the plant was living, it's just too massive. It'd be fairly challenging, especially fungi, like fungi here.
Like in that rifle and then B, there's more mushrooms there.
Yeah. And if you take
Let
the,
it go.
if you take the lid off this, all the spores.
Ugh.
And so I've had that, I've tried it where there's just a little tiny fungus in there. I'm like, oh, I can get the plant lid out, but yes, as soon as you lift the lid, there's that air current and it just wafts over everything. And so you subculture spores with it and it just doesn't work, so. So tissue culture is very common and getting more common as, scientists are learning how to work with tissue culture with all these different plants of commercial interest. So this one is a big deal in the cannabis world, hop latent viroid. It is a viroid, believe it or not, is a piece of RNA that infects plants and causes disease. Isn't that bizarre? It's just a piece of RNA that gets into the plant and can kill a plant if it's the right type. So it's not a virus. A virus has either DNA or RNA surrounded by this protein capsid. This is just a piece of RNA. That's all it is. And anyways, it's devastating the cannabis industry in terms of it's sweeping through it. I think they discovered it showing up in California. I have a former student who has a botany degree and she's working downstate in a cannabis growing company. And she contacted me last year saying, do you know anyone who's doing tissue culture work? We need to start doing tissue culture work so that we can get clean tissue. Because if they try to propagate the plant that has the viroid in it, it just comes with it because it's systemic. It goes through the whole plant. And so what they want to do is typically what happens if you have a virus in a plant, the only tissue where it seems to not be able to colonize is the shoot apical meristem for some reason. And so if you want to make sure that your tissue culture plantlets are disease-free, that's usually what you have to go for. It's the shoot apical meristem, and it's like performing microsurgery. You have to be really precise, and it's labor intensive. And so it's not commonly done because it's labor intensive, but she wanted to do that because she knew that the viroid had swept through their nursery. And so, is it in here? Where is? Yeah, the meristem tissue culture. So the shoot apical meristem is typically what they go for. And for some reason, viruses and viroids seem to not be able to get into that part of the plant. But do I have it on here? Yeah. Here. So this is what they're calling it the COVID of cannabis. That's how serious it is, this viroid. So has anyone heard about this? Okay. So it's a big deal. And I'll try to contact her, Danielle, and see what she's been able to accomplish on that.
Where's the orange stuff? Like, how's it get?
I don't know, I'm not a virologist, but it's just bizarre to me that it's just a piece of RNA and yet it can multiply, like make copies of itself and spread through the plant and cause all kinds of problems.
RNA is self-replicating.
Yeah.
So once it's
Yeah.
in there.
Viroids, as far as we know, viroids only attack plants. We have never found a viroid in a human situation or animal situation.
Oh, then all wants to be good.
Yeah. Knock on wood. Knock on wood, yeah. So, okay, so tissue culture is, like I said, gaining in popularity and not just in the cannabis industry, like African violet plants are very common household plant being sold. They do a lot of tissue culture work to get clones of those, but I mean, and on and on and on. All kinds of commercial plants are done this way. Okay. So there's also the idea that you might want to insert a gene into a plant that plant doesn't have. And so you need to think of a way to get that gene into the plant. And So one amazing way to do this is to use this particular bacteria, Agrobacterium tumefaciens. I'd like you to know that species, please. So Agrobacterium tumefaciens is a plant pathogen, and they've known about it. Plant pathologists have known about it for decades and decades. But then they were starting to studying it more carefully with more current technology, and they realized the way that it causes successful infection is that it inserts some of its own DNA into the plant chromosome. So if you've ever seen, do I have a picture? Oh no, I didn't put my picture in. If you've ever seen, if you're out hiking around here in town or even in the woods or in town, trees that have a big like tumor looking thing on the tree trunk, that's often caused by Agrobacterium tumefaciens. It's called a gall. The fancy word is gall, Gall. Ugh. Come on. Why won't it? Right. Oh, Gall, gall. I can't get my thing to write. Anyways, so what they're doing is the Agrobacterium infects a stem of a plant. And it then has this plasmid, it's called a T sub I plasmid. I stands for inducing, tumor inducing plasmid. So they actually refer to it as a tumor, which I find interesting, even though in the plant world, we say gall. But anyways, so here, what the pathogen does is it gets into the stem tissue, and then it gets into the cells, and then it inserts its plasmid into the chromosome of the plant. Then the plant starts expressing the genes that are on that chromosome. And one of the genes that it expresses is the auxin hormone is being overproduced in that area. There's an auxin gene on there. And so the gall starts to form. So you get this stimulus in the stem region, grow, divide, grow, but no differentiation. And so you get like this tumor looking thing on the stem. And, it's like, well, why would the bacteria want to do that? It's creating a home for itself. If you cut into that tumor and look at it, it's chock full of these bacteria. So the bacteria is pretty much telling the plant, make a little home for me. I'm going to hang out here. And then one of the other genes on the plasmid codes for a nitrogen containing compound that they make the plant make. So the plant starts expressing that gene. It starts to synthesize this nutrient that only the bacteria can use. The plant can't use it. So not only is the bacteria saying, make me a home. Oh, and by the way, make this nitrogen containing nutrient that I eat, but you can't. It's really an amazing little pathogen. And then the other thing is it usually doesn't kill the plant. It usually just hangs out in that gall or in that tumor long-term. And the only way that the plant really would die is if another disease comes along and weakens the plant enough, and then maybe the plant will die. But typically they don't die from this infection. Some of them are caused by this, but there's other things that can cause. Yes, there's fungi, there's insects. Yes. Yes, it doesn't go systemic typically. It tends to stay in the tumor tissue. So when they figured out that you can have this pathogen insert its plasmid into the plant chromosome, they're like, well, but that's what we want to do. We want to put a chromosome into this plant. And by the way, historically, it's really hard to transform a plant. So transformation means putting a gene of that you desire into the plant. That's really hard. And I'll explain why in a little bit. But so they're like, well, we can just use Agrobacterium for our desire to have a certain gene. So here, maybe this is, drought resistance or disease resistance gene that you want to put into a plant. And so you can work with the Agrobacterium in the lab. It's pretty easy to insert a section of DNA into the plasmid and then get the bacteria to take it up. And then you grow the bacteria to high levels, and then you expose the plant cells to this. So typically you're working in the lab, you're working in a liquid broth environment where you first have to grow up the bacteria with your gene of interest. And then you have the bacteria and the plant cells mixed together. And then you have to do it that way so that every cell will get this plasmid in it. That's the goal. And so what the other thing they've done is they've knocked out the genes that cause the tumor and all of that. So basically, they're just using agrobacterium as a way to deliver your gene of interest very easily into the plant. You make the agrobacterium do the hard work for you. And it's amazingly successful and pretty straightforward. The other really nice thing about agrobacterium is it has a broad host range. It can infect a lot of different species. So it's not just limited to a certain handful of species. So it's widely used. So here is what you could imagine happening. Here they're picking, so this plant is susceptible to a certain herbicide and they want to make it resistant, apparently. And so what they do is they take some cells from the plant and they will put it into a broth culture. And they have the agrobacterium in there. And they have the plasmid with their gene of interest, and they just mix them together. And they're looking for which cells did agrobacterium infect and get its plasmid in there. So what you do, if you're testing, you put your gene of interest, you want an herbicide resistant gene, then you usually tag it with like an antibiotic gene too. You put an antibiotic resistant gene in there as well. Because you have to be able to tell which gene or which plant cell got successfully infected. And so you have to have a way to figure that out. And so what you do is you usually use media that has an antibiotic in there, and anything that survives the presence of the antibiotic, supposedly, yes, they got the plasmid and will have the herbicide resistant gene in there too. And so it's all done in the lab. You have to use tissue culture techniques if you want every cell of the plant to have that gene of interest. You have to start with tissue culture. You can't start with a mature plant. You have to do tissue culture. So this is another idea too, just showing, and this is disease resistance now that they're looking. And here's the idea of, excuse me, the gene being put in into the plasmid, and then the marker is usually an antibiotic. And then you have, oh, they misspelled it, Agrobacterium tumefaciens, that shouldn't be an R in there. And so if the plant cell gets infected, here they're showing the bacteria inserting its plasmid into the chromosome of the plant. And then they put it onto agar that has that antibiotic. So anything that survives, they know, okay, if it survived and grew on that agar that has the antibiotic, then it did receive the plasmid, and it should be carrying the gene we want, which is disease resistance. Questions on that idea? Okay, well, so Agrobacterium tumefaciens. Oh, this is just for studying this, says it step by step what's going on here. So Agrobacterium tumefaciens is a really powerful workhorse in order to introduce genes of interest into plants and make it fairly straightforward. Sometimes though, you can't use Agrobacterium because it won't infect the plant that you're interested in. And so you have to get some other technique to work. But basically, when you're doing this, you want, if it's disease resistance that you're trying to create in that plant, you need to get a lot of copies of that gene made. And so that's one of the techniques that you do. Basically, you clone the gene, which just means you make lots of copies of it. in order for this to be successful when you're working with it in the lab. And so one way you could do that is again, using bacteria to be the workhorse. And so E. coli is commonly used for this. So you put your gene of interest into a plasmid, get E. coli to take it up, and then you just be nice to E. coli and let it grow and grow and grow. And as it's growing and dividing, it's making copies of your plasmid too. And then you end up with many, many copies of your plasmid, and then you can extract that and use it. And so very often we use just typical bacteria, and they're so quick to grow. You can get thousands of and millions of copies if you want of that gene. And then you've probably, if you've taken BI-218, you might have used PCR. That's another straightforward way. It's just a machine that is set up to take and make copies of your DNA. And that again would work really well. So that's another strategy that you can use to get lots of copies. And then once you get all those copies, you have a much higher success rate of introducing those into the broth culture with the plants cells in there. But there's a problem with plants in general. Mixing those plasmids in with a bunch of plant cells in that liquid culture, there's the cell wall that you have to deal with. The plant cell wall is formidable to try to get DNA past that and get it so that it'll get taken up and brought to the nucleus inside. And so that is a real challenge. And so one of the things they have to do is if they can't use agrobacterium to infect the plant, they usually have to figure out, okay, can we somehow create a situation where we can force the gene of interest or the plasmid or whatever past the cell wall, or maybe we can remove the cell wall very carefully and work with the cell like that. And so here are some techniques that they use in order to try to get some of these genes into the plant. So one is applying an electric field. So you usually do this in a liquid, and it's called electroporation. We don't have that here, like when BI-218, electroporation is not an option if you take that class, but essentially it pulses an electric field and it creates a temporary weakening of the cell wall where the DNA can then fit through well enough and get in to the cell and eventually to the nucleus. You could take your plant cells and treat it with a solution of polyethylene glycol, which again temporarily weakens the cell wall and allows the DNA to get in a little bit easier. Use a gun. I'll show you what I mean. It's called a gene gun. It's fun. I got to work with one when I was a PhD student. I'll explain that one in a minute. Microinjection, I'll show that, I'll picture that too. And then liposomes, I'll show you a picture of that. Just hang on. And then this figure is showing laserfection or optoinjection is what they are calling either one of those terms. But yeah, they use laser beams to get the plant cells, again, to be transiently, they often say that transiently permeable is how they phrase it. With that, so basically what they mean is we're relaxing and kind of temporarily relaxing the cell wall enough that we can get things in. And so here, all kinds of things can be sent into the cell so that's kind of one of the newest techniques that they have currently. All the other ones that I've listed have been around for quite some time and have some interesting impacts. So this is a gene gun. And so I didn't do it with plants, I did it with fungi, but fungi are also really hard to transform because they have a cell wall. And so they're also challenging. So you work again with cells and tissue culture media, and you actually have the bullets are tiny, of course, and they're coated with DNA. And so you actually get these little tiny pellets. You have to first get them all coated with your DNA of interest, and then you literally shoot it at the tissue. And the bullets will very quickly penetrate through the cell wall. And the cell can recover and fix that part that got a hole blown into it and recover and grow. It's bizarre, but it works. And they still use this biolistic particle delivery system. I like gene gun better. I think that's a little bit better, but
If you think about it, all guns are particle
Yeah.
delivery system.
I know, it's kind of funny. But yeah, it worked. We did it with a mycorrhizal fungus and it worked. Yeah, so it wasn't my project, it was a fellow grad student, but I had to get in on that. That was so cool. But here's you know, it actually kind of looks like a Nerf gun. Yeah, hot glue gun is a good one, yeah. So, but that's the idea here. So here they have down plant protoplasts, and that's even better. If you have a protoplast, what that means, I have a picture of, well, I'll show you in the next one too. If they say plant protoplast, that's specific. What they mean is they strip the cell wall off. I would like you to know that definition. So if you strip the cell wall completely off the plant cell, well, then you're left with the cell membrane and everything inside, then you call it a protoplast. And you can do that. I've had undergrads do that procedure on some plant cells and it works. Yeah, it's a delicate procedure and it's got a low success rate, but if you essentially have to treat it with the right concentration of enzymes that break down the cell wall. And then once you have the protoplast, it's much, much easier then to get, in this case, the bullet to penetrate in. So here again is a nice picture of protoplasts. And this is the idea of microinjection, kind of like in vitro fertilization with sperm and egg. Except this time you use your gene of interest that you inject into protoplasts. So these are all protoplasts, they have their cell walls. And microinjection does work. And then, of course, if you want to grow those up, you do tissue culture work. You get those transformed cells onto tissue culture media. Every cell will have that gene of interest in it because you started with one cell. And so you can get a whole plant transformed by this method, which is pretty amazing. So here they're showing the different parts of the plant too that you can source from, even the flower. So they're probably the earliest work with this idea was herbicide resistance in the commercial and the ag industry. They wanted a way to be able to spray their plants to get rid of weeds, but keep the crop of interest safe. And so herbicide resistance was probably the first genetic trait that was used to transform plants. So here you're seeing on the left of this image, you're seeing corn with they're resistant to the herbicide and you see there's no weeds around it. Whereas here on the other part of the plot, these are, you know, that's what you're faced with if you're trying to grow your crop of interest. Remember, the definition of a weed is any plant growing where you don't want it to grow. So even if this is lovely alfalfa, but that's not what you make your living off of. You make your living off corn. You don't want your alfalfa there stealing precious nutrients and minerals. So that's a problem. And so the idea here of herbicide resistance was the first big idea that using some of these techniques. Virus resistance, especially with some of the tropical crops, we see a lot of viruses love to attack. And so this is papaya plantations. And then here, looking at, we're running out of We're running out of usable soil, you know, good soil. Here's the idea of if you have high salt soil, it's too salty. They're trying to engineer plants to be okay with that. And so here they're looking at using, putting extra copies of a sodium ion pump so that that would get formed in the cell membrane so that the plant has extra copies so they have extra ability to pump out sodium. And it just allows them to survive in a high sodium soil, high salty soil. And so they're looking at these types of ideas too, of, how can we work with some of the marginal soil systems that we have left? And then you were probably too young to hear about this issue, flavor saver tomatoes. I think this happened before most of you were born. But this met with some resistance in the United States because of the genetic modification, and it's a plant part that you eat. And so, but basically, it was a way to resist spoilage during transfer. If you've ever eaten a tomato, you know how fragile they are. And trucking them thousands of miles across the United States, a lot of damage. And the loss is significant, financial loss from that. And so they created flavor saver tomatoes that would reduce their spoilage rates in transit. So there's things like that too. Okay, the next topic I want to get into, it requires a little bit of explanation and we're already out of time. So we'll finish this off on Wednesday and then start getting into fungi.